All things health and fitness, this is the Army Gymnastics Podcast.
The Army Gymnastics Podcast.
Discover a fitter, healthier you at armygymnastics.com.
Welcome, everyone, to the Deep Dive. Today, we're delving into something really personal, something that touches on hope, resilience, and, let's be honest, sometimes real challenges the journey of building a family.
It's a path many travel, and it can feel, well, overwhelming with information sometimes.
Exactly. And that's why we're here. We want to cut through some of that noise, look at the science, and give you clear, actionable insights on a really crucial topic: antioxidants and fertility, powering mitochondrial health.
Our goal is really to unpack the science, make it accessible, and help you feel genuinely informed about how cellular health, especially mitochondria and antioxidants, plays a role.
We might even uncover a few surprises, particularly about how incredibly resilient human eggs actually are. So let's start right at the source, the cell's engine room, the mitochondria.
Right, the famous powerhouses. They generate ATP, adenosine triphosphate.
Which is basically the energy currency for everything our cells do, right? From muscle movement to brain function.
Absolutely. It's generated through a process called oxidative phosphorylation, a very elegant, efficient system.
But like any engine, it has exhaust fumes, doesn't it? That's where these ROS come in.
Precisely. Reactive oxygen species, or ROS.
Yeah?
They're a natural byproduct of using oxygen to make energy. Tiny, very reactive molecules.
And they're not inherently bad. I think that surprises people.
Not at all in low amounts. Actually, they're vital. They act as signals, telling cells what to do. The key, like so many things in biology, is balance.
Okay, so balance is key, but things can get out of balance, leading to this thing called oxidative stress.
Exactly. When ROS production overwhelms the cell's natural antioxidant defenses, that's oxidative stress. Think of it like a factory's waste piling up because the disposal system can't cope.
And this damages the factory itself, in this case, the mitochondria.
Yes, it leads to mitochondrial dysfunction, or MD, as a double hit. The mitochondria produce less energy, less ATP, and at the same time, they actually start generating more oxidative stress.
A real vicious cycle, then.
A vicious cycle, precisely, and it impairs all sorts of cellular functions.
So how does this connect directly to fertility, to building that family?
The link is quite direct and, honestly, profound. Mitochondrial dysfunction is strongly linked to a decline in the quality of oocytes, the eggs.
Okay, lower quality eggs.
Which then leads to problems with embryo development, and ultimately it can really affect pregnancy outcomes, lower chances of success.
And you mentioned this isn't just about fertility.
No, not at all. MD is implicated in a whole range of conditions, neurodegenerative diseases like Alzheimer's, cardiovascular issues like atherosclerosis. It's a fundamental aspect of cellular health.
Wow. And even oxidative stress during pregnancy can have long-term effects on the child.
Research suggests just that, potential links to long-lasting cardiovascular effects in the offspring. So the cellular environment during development is incredibly important.
That really brings home the importance. Now, what about fertility treatments themselves? Things like IVF or RRT, can they contribute to this oxidative stress problem?
It's an important point. While art is life-changing, the procedures themselves can inadvertently increase ROS levels. It's not a flaw, really, but a consequence of taking these delicate cells out of the body's tightly controlled environment.
Into the lab setting, the in vitro world.
Right. And in the lab, you get both indirect ROS, the cell's stress response to the new environment, and direct exogenous ROS from factors in the lab.
Like what kind of factors?
Well, things you might not immediately think of. Oxygen levels, for instance. The air in the lab has much higher oxygen than inside the body.
Which the eggs aren't used to.
Exactly. Also, temperature fluctuations, even slight ones, exposure to bright light during handling, the specific chemicals in the culture media they grow in, and even the freezing and thawing process, cryopreservation.
So lots of potential stressors, and the natural protection isn't there.
Largely absent, yes. The body has this amazing, complex system of antioxidants in fluids like follicular fluid. In the lab dish, that natural buffering system isn't fully replicated, making the gametes more vulnerable.
This leads us right to the core question then. Can taking extra antioxidants help? Can supplementation actually improve egg mitochondrial function and boost fertility?
That's the million-dollar question, isn't it? Antioxidants are molecules that counteract ROS. Our bodies make them, but can adding more, through pills or in the lab, make a real difference?
How are doctors trying this now? What are the main approaches?
Two main ways, generally. First, Oral supplementation, taking antioxidants by mouth, hoping to improve egg or sperm quality before treatment even starts, kind of prepping the body.
Okay, taking supplements. And the second way
Is adding antioxidants directly into the culture media, the liquid the eggs or embryos grow in the lab. This aims to protect them right there in vitro, especially during processes like IVM, which is maturing eggs in the lab, or just generally counteracting the stress of the IVF environment.
Right. So does it work? What does the big picture evidence say? Because you hear a lot about antioxidants.
Well, here's where it gets nuanced. There are promising results, especially from animal studies or some smaller human trials. But the big, high-quality reviews, like the Cochrane reviews, they describe the evidence for oral antioxidants improving live birth or clinical pregnancy rates in women undergoing fertility treatment as generally low quality.
Low-quality evidence, that doesn't mean they don't work, just
Just that we don't have enough strong, consistent proof from large, well-designed human trials yet. The studies are often small, use different doses, different populations. It makes it hard to draw firm, universal conclusions. The jury is still somewhat out on widespread use.
Okay, that's a really important caveat. But it doesn't mean we shouldn't look at specific ones where there might be stronger signals, right?
Absolutely not. We need to look at the specifics. Today we'll focus on a few key players: coenzyme Q10, resveratrol, melatonin, folic acid, and vitamins A, C, D, and E.
Great. Let's start with CoQ10. It gets a lot of buzz in the fertility world.
It does, and for good reason. CoQ10 is really interesting because it plays a dual role. It's literally part of the machinery in the mitochondria that makes ATP the electron transport chain. So it's fundamental to energy production.
Okay, part of the engine itself and the antioxidant part.
Right. Its reduced form, ubiquinol, is a very powerful antioxidant.
Yeah.
It protects cell membranes, especially from fat damage, lipid peroxidation, and it even helps recycle vitamin E, a real multitasker.
And there's an age connection here, isn't there? Which is obviously very relevant for fertility.
A strong one. CoQ10 levels in our bodies naturally decrease as we age. And this decline mirrors, quite closely actually, the age-related decline in fertility and the increase we see in embryo aneuploidy, abnormal chromosome numbers.
That correlation seems pretty tight. What about studies? Animal studies often give us clues.
They do. And animal studies on CoQ10 are quite positive.
Yeah.
Supplementation in aged mice and rats has shown it can improve mitochondrial function, improve egg quality, and even seem to prevent or slow down ovarian aging in those models.
Wow, slow down the clock in mice.
In essence, yes, in those specific studies. It also seems to benefit the cumulus cells, those support cells around the egg, which are crucial. In aged mice, CoQ10 help these cells work better, use glucose better. And we see similar age-related declines in CoQ10 synthesis genes in human cumulus cells.
Okay, so promising animal data. How does that translate to humans? Often more complicated, right?
Usually, yes. It's a mixed picture. On the plus side, we do see that higher CoQ10 levels in the follicular fluid, that fluid around the egg, are linked to better quality embryos and higher pregnancy rates. So there's a positive association there.
Association, but not necessarily causation proven yet.
Exactly. Now, there was one specific study in older women, 35 to 43, taking 600 milligrams a day for 2 months. It showed a really interesting trend towards lower aneuploidy rates in their embryos.
A trend, so not statistically significant.
Unfortunately, no. The study ended early and was quite small, so it couldn't reach definitive conclusions. It suggested maybe longer treatment or higher doses might be needed. But mostly, it highlighted the need for bigger trials.
Okay, so aneuploidy prevention is still a maybe. What about women with poor ovarian reserve, another big challenge?
Yes. In younger women, but with poor ovarian reserve, that same dose, 600 milligrams a day for 2 months, did show significant benefits. They retrieved more eggs. The fertilization rates went up quite a bit, from like 45% to over 67%, and they got more high-quality embryos.
That sounds pretty good. More eggs, better fertilization.
It does. But, and this is crucial, that particular study did not find a significant difference in the downstream outcomes, clinical pregnancy, miscarriage, or live birth rates in that specific group.
So it helped at the early stages, egg numbers, fertilization, but didn't necessarily translate to a baby in that trial.
In that trial, for that group, correct. It shows the complexity. Improving one metric doesn't always guarantee the final outcome, or maybe the study wasn't powered enough to see that difference. It's an important distinction.
Very important. What about PCOS, polycystic ovary syndrome? Often involves metabolic issues.
Yes, CoQ10 showed some benefits there, too, especially for women resistant to clomiphene, a common fertility drug. It helped increase the number of larger, potentially responsive follicles.
Okay. And any benefit during IVM, the in vitro maturation?
Yes, a 2020 study was interesting. It looked at IVM for eggs from aged women. Supplementing the IVM culture with CoQ10 significantly boosted maturation rates and, quite remarkably, reduced aneuploidy rates after meiosis in those aged eggs.
But not in eggs from younger women.
Correct. The benefit seems specific to the aged eggs in that study, suggesting it might target age-related vulnerabilities.
So pulling it all together for CoQ10, what's the verdict? Is it worth considering?
Well, a large meta-analysis, pooling data from multiple studies, did find that oral CoQ10 as an IVF pretreatment was associated with increased clinical pregnancy rates, a significant jump, actually nearly doubling the rate in some analyses.
That sounds very positive.
It is. However, the studies included were very different. Different patients, doses, durations, high heterogeneity, we call it. And critically, the analysis didn't have enough statistical power to show a significant difference in live birth rates or miscarriage rates, not enough consistent data on those ultimate outcomes.
Okay, so the final word on CoQ10.
It's definitely promising, is readily available, generally safe, not too expensive, but we still need those definitive, large, well-designed trials to really nail down its effect on live births across different groups. It's a strong contender, but the final proof is still pending.
Makes sense. Okay, let's move to another one that gets attention, often for anti-aging. Resveratrol, found in grapes, red wine.
That's the one, a natural polyphenol. And on paper, it looks great for fertility, too. Antioxidant, anti-inflammatory properties.
That's like a winner. What have studies shown? Animal models first, perhaps?
Animal models are quite positive. It shows potential for diminished ovarian function, PCOS, endometriosis. It seems to work partly by activating SIRRT1, which helps protect mitochondria, and SIRRT1 levels are often lower in aged oocytes, so activating it seems beneficial.
So it helps protect the mitochondria. What else?
Studies in mice showed it could reduce the rate of follicle death, increase the ovarian reserve, basically prolong the ovary's lifespan. Mice treated long-term had ovaries looking much younger, with better quality eggs.
That sounds amazing for ovarian health.
It really does. It helped with various types of ovarian dysfunction in animals, improve maturation, embryo development. It seemed to reduce ROS and boost ATP. Looked like a miracle compound for the ovary.
But you mentioned a catch earlier.
Ah, yes, the big catch, the crucial catch. While it seems great for the ovary and the egg, resveratrol has a potentially significant negative effect on the endometrium, the lining of the uterus. Its anti-inflammatory properties, which are generally good, might actually inhibit decidualization. That's the essential process, where the uterine lining changes to become receptive to an embryo. It's actually an inflammatory-driven process.
Wait, so the anti-inflammatory effect is bad for implantation?
Potentially, yes.
Yeah.
By dampening that necessary inflammation, it might make the lining less receptive, preventing the embryo from implanting properly.
Wow. So you could have a great egg, but it can't stick. That's quite the downside.
It's a major dilemma. And unfortunately, human studies seem to bear this out.
Oh, dear. What did they find?
Well, on the one hand, resveratrol did improve egg quality in lab studies with aged human eggs, better maturation, healthier-looking mitochondria, fewer chromosome issues, and it helped reduce androgens in women with PCOS. All good for the egg-ovary side.
Okay, positive signals there.
But a key retrospective study looked at women taking oral resveratrol, about 200 milligrams a day. And the findings were concerning. They saw significantly lower clinical pregnancy rates dropped by almost half, and significantly higher miscarriage rates more than doubled after embryo transfer, even when good quality embryos were transferred.
Oh, wow. That strongly suggests the endometrial effect is real and problematic in humans.
It really points that way. The negative impact on the lining likely outweighed any benefit to the embryo itself in those cycles.
So is resveratrol just a no-go then? Or is there a way around this?
There are proposed workarounds. Resveratrol has a pretty short half-life in the body, maybe 3 to 9 hours. So one idea is to simply stop taking it around the time of ovulation.
Let it clear the system before the lining needs to become receptive.
Exactly. Allow levels to drop before that critical implantation window opens. The other strategy is to use a freeze-all approach during IVF.
Meaning, create the embryos while taking resveratrol, get the egg benefits, but freeze them all.
Right. Then transfer them in a later cycle, a frozen embryo transfer cycle, when the patient is not taking resveratrol. That way, you separate the ovarian benefit from the potential endometrial harm.
Clever. A way to potentially get the best of both worlds, but it requires careful management.
Very careful management and planning, yes.
Yeah.
It highlights how complex these interventions can be.
Definitely. Okay, let's shift gears to melatonin. Most people think sleep aid, right?
They do, but it's a really potent antioxidant too, and much more involved in reproduction than many realize. It helps modulate the main hormonal axis, the HPG axis, and it directly reduces oxidative stress within the follicle.
Directly protecting the egg's environment.
Yes. Some even suggest follicular fluid melatonin levels could be a biomarker for egg quality or IVF success. And, like CoQ10, melatonin production declines with age.
Another age-related decline link. Does it help with ovarian aging in studies?
Animal models suggest it can. Melatonin treatment in older animals has been shown to reduce signs of ovarian aging, improve egg quality and quantity, and even increase litter size.
So preserving fertility potential in animals, what about humans?
In humans, there's evidence it can offer broad benefits for infertility, including for women with PCOS or endometriosis, conditions often linked to oxidative stress. It might help regulate cycles and potentially protect the early embryo.
And specifically for egg quality, has it shown benefits there?
Yes, some key studies have. One found that 3 milligrams a day significantly improved egg quality in women who'd previously had IVF failure due to poor eggs. They saw fewer degenerated eggs and lower oxidative stress markers in the follicular fluid.
Fewer bad eggs, less stress around them. Did it help them fertilize better too?
It seemed to. Melatonin appeared to help with the final maturation process and significantly boosted fertilization rates in women who historically had trouble with that step. The increase was quite dramatic compared to the control group in that study. Other trials also found it increased the number of eggs retrieved and the number of mature eggs.
So more eggs, better quality, better fertilization. Sounds pretty positive overall. Any conflicting data?
Unfortunately, yes. Consistency is often the challenge. A well-conducted randomized trial in 2018 found no significant difference in any major IVF outcome. Eggs retrieved, mature eggs, fertilization, embryo quality, pregnancy rates, live birth rates between the melatonin group and the placebo group.
So one trial shows clear benefits, another shows none. Why the difference?
It's hard to say definitively. Different patient populations, maybe different underlying reasons for infertility, subtle differences in protocols. It just highlights that we need more research to clarify who might benefit most and under what circumstances.
Was there any specific group where it seemed more consistently helpful?
There was a study in 2019 focusing on women with unexplained infertility. They found these women often had lower melatonin levels. Supplementing with melatonin, either 3 or 6 milligrams, seemed to restore the oxidative balance, improve their egg quality, lead to more transferable embryos, and ultimately better RT success rates compared to similar women without the supplement.
Okay, so maybe particularly helpful for that frustrating, unexplained category. Interesting.
It suggests a potential targeted benefit, yes. But overall for melatonin, like CoQ10, it's promising in many ways. Yeah. But the evidence isn't uniformly conclusive across the board yet.
Right. Okay, let's turn to the vitamins. Starting with vitamin A. We know it's important generally, but for fertility.
Vitamin A, including retinol and carotenoids, is a known antioxidant. But it also plays loads of fundamental roles in the reproductive system, follicular growth, making steroid hormones, egg maturation, early embryo development. It's really essential.
Involved in almost everything.
Pretty much. Lab studies show adding it to IVM media can help eggs mature better, partly by improving mitochondrial health and reducing apoptosis or cell death.
And in humans, is there a direct link shown between vitamin A levels and egg quality?
Yes. Higher levels of its active form, ATRA, in the follicular fluid are linked to the best quality embryos and better fertilization rates. So adequate levels seem crucial for the egg to mature properly.
But is it the antioxidant effect specifically that's doing the heavy lifting here?
That's the big question we can't answer yet. Because vitamin A does so many other essential things for ovarian function, we don't know if the fertility benefits seen are primarily due to its antioxidant action or its other roles. And we lack human trials specifically testing vitamin A as an antioxidant supplement to improve egg quality.
So vital for reproduction, but the antioxidant mechanism for fertility benefit isn't isolated or proven through supplementation trials.
Got it. What about folic acid, vitamin B9? Everyone knows about taking it before pregnancy.
Absolutely. It's standard advice mainly to prevent devastating neural tube defects like spina bifida. And folate deficiency is linked to problems in animals like impaired ovulation or more degenerated follicles. And in humans, it's associated with poor ovarian function and lower pregnancy rates if levels are low.
So definitely important for a healthy pregnancy and maybe even getting pregnant. Again, is the benefit related to its antioxidant properties? Well, it might play a role in egg development and early embryogenesis, partly through antioxidant actions, But folate is also critically involved in so many other things, especially DNA synthesis and repair, methylation. It's hard to tease out just the antioxidant part.
It's doing too many important jobs to isolate one function easily.
Pretty much. And the human trial data, specifically looking at supplementation for fertility outcomes, beyond the neural tube defect prevention, is contradictory.
Contradictory? How so?
One study linked higher folate supplement intake to better implantation, clinical pregnancy, and live birth rates. But another found no significant difference in pregnancy or live birth rates based on blood folate levels or supplement use. So the evidence specifically for boosting fertility via its antioxidant mechanism through supplementation is inconsistent and lacks dedicated trials. Standard recommendation stands for NTD prevention, but not strongly proven as a specific fertility antioxidant booster.
Okay. Moving on to vitamin C, ascorbic acid, classic antioxidant, right? Scavenges free radicals directly.
Textbook antioxidant, yes. And it did show some benefit in animal studies, like improving the quality of frozen thawed embryos.
Suggesting a protective effect, but in standard IVF culture.
There, it didn't seem to make much difference to maturation, fertilization, or development in those animal studies. And more importantly, human IVF trials haven't shown significant benefits either. No major differences in implantation or pregnancy rates, even in women with endometriosis where oxidative stress is thought to be high. And this was despite vitamin C levels actually increasing in the women taking the supplements.
So even though levels went up, the key fertility outcomes didn't change significantly. Another case where being an antioxidant doesn't automatically translate to improved IVF success in trials.
It seems so. It underlines that the biological system is complex. Just adding more of one thing doesn't always yield the expected result.
What about vitamin D? It's getting a lot more attention for fertility lately.
It is. Low vitamin D levels are increasingly linked to poorer reproductive outcomes, things like PCOS, maybe IVF success. But the evidence is still considered, well, not entirely conclusive. There's a lot of active research.
Well, what about its antioxidant role specifically in eggs?
That's the interesting gap. We know vitamin D has antioxidant properties, and it can improve mitochondrial dysfunction in other body tissues. But its potential antioxidant effects, specifically in human eggs or sperm, it simply hasn't really been investigated properly yet.
So a potential player, but the specific antioxidant angle in gametes is uncharted territory for now.
Exactly. A big area for future research.
Okay, last one in this group. Vitamin E, known for protecting cell membranes.
Yes, alpha-tocopherol. Its main job is protecting fatty cell membranes from oxidative damage, breaking the chain reaction of lipid peroxidation.
And the evidence. Animal studies.
Some benefits seen there. Adding it to culture media helped blastocyst development, and combining it with vitamin C seemed to help prevent ovarian aging in mice, suggesting a synergistic effect perhaps.
And humans. What's the link between vitamin E levels and fertility outcomes?
It's nuanced. Studies have found correlations, specific ranges of vitamin E in follicular fluid linked to better egg maturation, and certain serum levels linked to getting more high-quality embryos. So having the right level seems important.
Not too high, not too low.
Potentially. But here's a key point. A trial that combined vitamin E with vitamin D for women with PCOS did show improved pregnancy outcomes. However, the researchers concluded these benefits were not associated with an antioxidant mechanism.
So a benefit was seen, but they attributed it to something other than vitamin E acting as an antioxidant in that context.
Correct, which means we still need more research to figure out if vitamin E's antioxidant effect specifically directly improves oocyte mitochondrial function or overall fertility. The correlation is there, but the causal antioxidant link isn't firmly established by intervention trials.
It really underscores a theme, doesn't it? These vitamins are essential, many are antioxidants, but proving that the antioxidant function is the specific key to unlocking better fertility outcomes through supplementation is often where the evidence gets murky or is still lacking in humans.
Precisely. The body is complex, and these molecules wear many hats. Now, this leads us perfectly into another critical area, moving beyond just supplements, the fundamental battle for DNA integrity in sperm and eggs.
The genetic blueprint itself, this feels absolutely core.
It is. Both sperm and egg are constantly under siege from stressors that can damage their DNA, proteins, lipids, compromising their ability to create a healthy embryo.
And sperm and eggs handle this challenge very differently, don't they? A real tale of two gametes.
Dramatically different strategies. Let's start with sperm. Males produce billions of them throughout life. It's continuous production involving countless rounds of DNA replication.
Lots of copying, which means more chances for errors.
Exactly. This leads to a gradual buildup of mutations in the sperm-producing cells over time, especially with age. It's estimated that each extra year of paternal age adds about 1 to 3 new mutations, de novo mutations, to the offspring's genome.
1 to 3 new mutations per year of age. That's significant. Now, contrast that with the egg.
The female situation is mind-bogglingly different. Women are born with all the eggs they'll ever have, maybe 1 to 2 million. This number plummets by puberty and keeps declining. Crucially, these eggs sit paused, in meiotic arrest, for decades, up to 50 years.
Waiting patiently for half a century, how on earth do they stay healthy and viable for that long?
That's the incredible biological puzzle. They have to maintain integrity for decades, unlike continuously produced sperm. This led to a major breakthrough discovery about how they manage it, this standby battery mode.
Right. This overturned previous ideas, didn't it? What did scientists think eggs were doing?
Based on mouse studies, the assumption was human eggs would constantly be cleaning house, ramping up protein degradation, actively fighting off damage, very metabolically active.
But that's not what Dr. Boak's team found in human eggs.
Not at all. They found human oocytes do the opposite. They actually dial down their waste disposal systems, proteasomes, lysosomes, and critically, they dampen their metabolism way down. It's a strategy of quiet preservation, minimizing wear and tear over the long haul.
Less activity to preserve themselves. Genius. And what about ROS, the oxidative stress we talked so much about?
This was maybe the most stunning finding. They described it as pitch black. Human oocytes showed virtually no detectable ROS activity, unlike almost any other cell.
No ROS. How?
They found human oocyte mitochondria essentially lack complex 1, which is the main entry point for electrons in the energy production chain, and also the main site of ROS leakage, like exhaust fumes.
So they bypass the leaky part of the engine.
Exactly. They use complex 2 instead, which is less efficient at making ATP, but produces far, far less ROS. They generate just enough energy to survive in this standby mode without creating the damaging byproducts. An incredibly elegant solution for longevity.
Wow. So instead of fighting ROS with antioxidants, they just don't make much ROS in the first place.
Precisely. It's a proactive prevention strategy built into our core metabolism.
And they do a bit of spring cleaning right at the end, too.
Yes. Live imaging showed that as the egg matures just before ovulation, it literally ejects some components, like lysosomes, into the surrounding space. And other things like mitochondria move to the edge. A final clear-out.
What does this mean for fertility treatments, then? If eggs are naturally quiet, should we be trying to boost them?
That's the profound implication. Maybe the best approach isn't trying to supercharge the egg with supplements, but rather respecting and supporting its natural, quiet, low metabolism state. Perhaps less is more when it comes to preserving egg quality. Don't mess with nature's hack.
A really fundamental shift in perspective. But even with these amazing strategies, damage can still happen, right? Let's talk about the types of DNA damage. You mentioned defective protamination in sperm.
Right. Protamines are proteins that replace histones to package sperm DNA incredibly tightly. If this packaging process goes wrong or the ratio of the two types of protamines is off, the DNA is left more vulnerable, more prone to breakage, like a poorly packed suitcase.
Okay. And abortive apoptosis. Sounds ominous.
It means some sperm precursor cells that were actually signaled to die off via apoptosis manage to survive and develop into mature-looking sperm, but they carry fragmented DNA from that incomplete death process. They look okay, but carry hidden damage.
Damaged goods that slip through quality control. And oxidative stress is still a factor.
Always a factor. It damages DNA bases, especially creating 8-oxyG, which leads to specific types of mutations when the DNA is replicated later in the embryo, a constant threat.
And ironically, RT procedures themselves can cause some damage, iatrogenic damage.
Yes, it's a known risk clinics work hard to minimize. Things like light exposure, chemicals in the air, centrifugation, removing the natural protective seminal or follicular fluid,
Right.
all can increase ROS. And cryopreservation, freezing, and thawing can also cause some oxidative stress.
It's a delicate balance. Now, you mentioned single versus double-strand DNA breaks. Why is that distinction so important?
Hugely important. Single-strand breaks, SSBs, where just one side of the DNA ladder is cut, are often repairable by the egg after fertilization. But double-strand breaks, DSBs, where both sides are severed, are much more serious.
More serious how?
A high load of DSBs can simply overwhelm the egg's repair capacity. This can lead to the paternal DNA not replicating properly or even being degraded, causing the embryo to stop developing. Plus, DSBs have a higher risk of being repaired incorrectly, leading to harmful mutations. So DSBs are a much bigger problem for embryo viability.
That makes sense. And finally, these de novo mutations, the silent genetic threat, remind us what they are.
They're new mutations that appear in the offspring but weren't present in the parent's body cells. They arise in the sperm or egg.
And there's that strong paternal age effect.
Yes, significantly stronger from the father's side. Remember, 1 to 3 new mutations per additional year of paternal age versus only about 0.24 for maternal age. It's because of all those extra rounds of DNA replication in sperm production over a man's lifetime. More copying equals more chances for errors.
And the impact of these DNMs.
They are a major cause of severe early-onset developmental disorders, autism, intellectual disability, congenital heart defects, also linked to later-onset neurological or psychiatric conditions. Understanding them is critical, especially as RT sometimes bypasses natural sperm selection.
Okay, so a lot of potential sources of damage. It really highlights how vital DNA repair mechanisms must be. Our cells fight back, right?
Absolutely. Germ cells have a whole toolkit of DNA repair pathways, NER, MMR, BER, HR, NATJ, constantly working to fix damage and maintain the integrity of the genome. It's an ongoing battle.
What about sperm specifically? Can they repair their own DNA once they're mature and packed so tightly?
It was thought they couldn't, but we now know they have a limited but functional base excision repair, BER, pathway. Specifically, they have the OGG1 enzyme to snip out oxidized bases.
So they can start the repair.
They can start it, yes. They remove the damaged base, but they can't complete the next steps. The egg has to finish the job after fertilization, repairing the gap left behind.
Wow, that's amazing teamwork again. The egg takes over the repair process. How good is the egg at this, given it's been waiting for decades?
The egg's repair capacity is remarkable. It has to be, to maintain its own DNA integrity for so long. And in the early zygote, before the embryo's own genes turn on around the 4-cell stage, DNA repair relies heavily on the machinery the egg provides. It's very much a maternal trait initially.
The mother's contribution is crucial for that early repair, this fortuitous complementarity you mentioned.
It's beautiful biologically. The egg can fix a lot of the damage the sperm brings in, especially those SSBs and the incomplete BER sites. Some repairs genuinely need both gametes working together.
But the egg's capacity isn't infinite.
No, it has limits. High levels of sperm DNA damage, especially those nasty DSBs, can overwhelm the system. That leads to poor embryo development or loss, the late paternal effect. The egg does its best, but it can be overloaded.
And does the mother's age affect the egg's repairability?
Unfortunately, yes. Just as egg quality declines, so does its DNA repair efficiency with age. This is likely due to lower levels of the mRNA transcripts needed to make the repair proteins. So older eggs are potentially less able to fix damage, compounding the age effect.
So we have these amazing repair systems, but there's still a big unanswered question for couples undergoing fertility treatment, isn't there?
There is. Current tests can tell us if sperm have fragmented DNA, but they cannot tell us if that specific damage is actually repairable by the egg. That's the missing link predicting repairability. We can see the problem, but not necessarily the prognosis for repair.
That's a huge gap. So wrapping this all up, it's been an incredible journey through mitochondria, antioxidants, DNA damage, and repair. The interplay is clearly critical for fertility.
Absolutely. It's this intricate dance between energy production, managing oxidative stress, and maintaining genetic integrity.
And while antioxidants show promise, especially for maybe improving mitochondrial function or reducing stress during ART, the direct proof, particularly in human eggs leading to more babies, is still something researchers are actively working on for many of these supplements.
Exactly. The research is ongoing, and we need more high-quality evidence to guide clinical practice definitively for many of these compounds, especially regarding their specific effects on mitochondria in human oocytes.
And understanding the egg's own incredible biology, that quiet, low ROS standby mode, seems just as important, maybe more so, than just trying to add things in.
It really shifts the perspective. Supporting the egg's natural strategy might be key. Developing treatments that respect that unique biology is likely the future.
And recognizing that RT, while amazing, bypasses some natural checks and balances, makes understanding gamete health and repair even more vital for ensuring the best outcomes and reducing risks.
Precisely. Better understanding leads to better care and, hopefully, better success rates for families.
It leaves us with a thought, doesn't it? In a world so focused on intervention, maybe the most powerful fertility hacks aren't about adding more, but about deeply understanding and gently supporting the quiet, inherent resilience built into the very start of life.
A very profound thought to end on. It's about working with biology, not just on it.
We really hope this deep dive has illuminated some of the complex science behind antioxidants, mitochondrial health, and fertility for you. Thank you for joining us.
Thanks for listening. For all things health and fitness, subscribe to our newsletter today. Find out more at armygymnastics.com.